Welcome to the Chapter 5, Chapter 12, Section 5 podcast. And this is on linkage and gene maps. And the first question we're going to ask is, what structures actually assort independently? If you remember correctly, Mendel said that that genes assort independently, but the evidence didn't quite show that when people did further genetic testing. So Thomas Hunt Morgans was a guy who did work on fruit flies, and fruit flies are really easy to use, as we've talked about. They breed quickly, they're easy to keep, they're easy to take care of, and by using them, he came up with a theory on what's called linkage. What he found that some of the genes that the Drosophila had, the fruit fly had, appeared to be linked together. Meaning, if you inherited one trait, you were very likely to inherit another trait from the same that's a different gene, but somehow you seem to get them together, and this seemed to violate the principle of independent assortment. So what they did was they grouped the genes, the linked genes, meaning the ones that were inherited to be that were likely to be inherited together, linked them into 4 linkage groups, and each linkage group assorted independently, but all the genes in one group were inherited together. Now, what this led to was the understanding of a chromosome. Chromosomes are physically linked together, and it's a group of linked genes. So what Morgan concluded that Mendel's principle of independent assortment holds true. However, chromosomes assort independently, not individual genes. And Mendel didn't quite figure out that some genes were linked together simply because he only looked at 7 traits, and as it turns out, the 7 traits that were on the pea plants, if I remember correctly, 5 were on their own chromosome by themselves, so they would sort independently, and the other 2 were very far away from each other on the same chromosome, and we'll show why that's significant in a moment. So what happens during crossing over, during meiosis, is the genes that were on the same chromosome, that had been on the same chromosomes, are on homologous chromosomes. So sometimes they separate them, and when they separate the linked genes, it produces new combinations of alleles. So if you look down here, you can see this section of green right here with the little a to the little b is going to cross over with this section of blue on the homologous chromosome with the capital A and the capital B. And as you can see, here's the result. This section right here is green, this section right here is blue, and they exchange those pieces of chromosomes. So what Albert Sturtevant, who was a student of Morgan, ironically, reasoned that the farther apart 2 genes were, the more likely they were separated by crossover during meiosis. And if you look at this right here, you can see that gene A and gene E are very far away from each other on the chromosome, so they're more likely to be separated than, say, gene A and gene B, which are right next to each other on the chromosome. There's more likely a separation going to occur somewhere between A and E than there is between A and B. So what they've found is recombination frequencies allow geneticists to determine the distance between genes on the chromosome. And what this allows is for people to create a gene map. Now, remember, if Hunt Morgan's group found there were 4 linkage groups, guess how many chromosomes there are in a fruit fly? There are 4. And so now they know where each gene is on the Drosophila chromosomes. So if the 2 genes are close together, the recombination frequencies should be low, since the crossovers are going to be rare. And if they're far apart, recombination rates should be high, meaning they're more likely to be separated from each other. Here's 3 chromosomes, or 2 of the chromosomes on the fruit fly That's chromosome. I'm sorry. That's chromosome. This is 2 chromosomes. They're both chromosome 2. Just showing you the map, and it shows you the relative distance from each other on the chromosome. And this is just all of them together. It's kind of a redundant slide. So, all right. So here, let's draw some analogies here. If you were to map the Earth and map the cells and chromosomes, you could think of the Earth as a cell, and then within the Earth you have countries, and each country would be a chromosome, and then within the country you might have a state, which would be a piece of a chromosome. Then you might have a city, which would be a gene, and then the people within the city might be nucleotide base pairs.